All right, welcome to our first in-person seminar in a while. Glad to have everyone here, and thanks for making the time and coming over. So we're going to be recording this talk. It's not live streamed, but we'll record it and put it on our YouTube channel afterwards, just for people who weren't able to make it for whatever reason. So lucky you guys being here, you can review any of the information later if you choose to. So I'd like to give our land acknowledgement to start. We'd like to respectfully acknowledge the land of Newfoundland as the ancestral homelands of the Mi'kmaq and the Otak. We also recognize the Inuit of the Natsivut and the Naktavut and the Innu of the Natsnick and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all people of this province as we work towards true reconciliation and honor this beautiful land together. We offer this land acknowledgement to recognize Indigenous peoples' connection with the land we live and study on. As students, acknowledging the Indigenous history and present of the places we study is an important contribution we can make towards conciliation. And voicing this statement at the beginning of each seminar is a start, and we hope it provides a prompt for reflection in our audience and ourselves, reminding us to learn with open minds and hearts so we can better contribute to communities and the world through our studies. We welcome feedback on our words as we continue to learn and the perceptions and needs around this type of acknowledgement evolve. So today we have the supreme pleasure of hearing a talk from Dr. Craig Purchase. Dr. Purchase is a professor of biology at Memorial. He did his master's at Memorial and then took a little trip out to Ontario to do a PhD at the University of Toronto. Dr. Purchase has experience in the private and public sector before coming into academia, and he is involved currently with Science Atlantic, which has an upcoming conference, I think next weekend, and as well as the Salmon Association of Eastern Newfoundland, and is the president of the Canadian Aquatic Resources section of the American Fisheries Association. Dr. Purchase is an evolutionary ecologist focusing on fish reproductive biology, looking at intra and interspecies interactions, as well as abiotic interactions, and studies interactions from behavioral all the way to genetic. So a really broad, interesting subject. And without further ado, wasting no more time, take it away, please.
Well, thank you, and it's nice to be in a room with people again, and thanks for the invitation for the seminar. I was thinking about it afterwards. Although I've given many departmental seminars, this is the first seminar I've given this department since I interviewed for my job, which was just over 15 years ago, and Andrew and Yolanda would have been there. So yeah, this is the first time since. So, if we think about life on this planet, if we had to boil this down to a couple little things, one slide that could encompass much would be this. Because other than maybe something Andrew works on, for example, most life on the planet is eukaryotic, and this simple slide encompasses eukaryotic life, where we have a biphasic life cycle, we have a diploid phase and a haploid phase, and these vary a little bit depending on if you're a plant, etc. I'm going to focus my talk on animals, and in animals, one complete circuit around the cycle represents a generation. And if you haven't picked up on the pond, your nuts are diploid and your sperm are haploid. We think about generations as very distinct things, but they're actually not. So if we go back through time, all generations are just connected in a swirl like this across haploid, diploid, haploid, diploid, haploid, diploid. It's an unbroken chain through time. And that has implications for how we think about one generation influences the next. Some of you have seen this cartoon before, and I'm going to spend some time on this. I made a version of this a few years ago as a way to try to conceptualize where selection happens across the biphasic life cycle. So I'm going to walk through this a little bit, ignore the stuff in the boxes for now, but in an animal, a generation starts at fertilization. So the development of a zygote is the beginning of a new generation. And with cell division, you get the development of an embryo, and then basically all animals look the same, and then cross the juvenile phase and to some adults. And depending on the taxa of animal, from a zygote to an adult could take days or weeks, or it could take decades, or it even could take centuries. But the process is identical in all of those situations. Adults produce gametes, which are the haploid phase of a biphasic life cycle for animals. They're typically quite short-lived, and when those gametes fuse, you basically get the creation of a next generation. Now, across that cycle, selection can happen at different parts. And so this was an attempt to try to categorize that into broad-reaching things. This is illustrated for animals with some examples, but for example, with Jalissa's help, I've got this contextualized and developed with plants, and we have a paper on this that we haven't published yet, but it is available as a preprint, but kind of incorporates terminology and everything for plants into this process. But I'm just going to stick with animals for the simplicity of the story. So if we start here at the beginning of a generation, the first opportunity for selection is basically Darwinian natural selection. So it's natural selection in the diploid phase, and really, you know, if one of Eric's students are measuring caribou calf survival, this is what they're doing. And if we think about it in the big picture, what natural selection in the diploid phase actually is its zygote to adult survival and the ability to produce and release gametes. And it doesn't matter if that zygote to adult survival takes a week. I don't know, how long does this Drosophila take? 2 weeks? I don't know, something probably in that scale. Or 150 years, like a Greenland shark. Conceptually, it's all identical. So we can think about this as, of what was produced, who lives to reproduce? But if you've lived to reproduce, not everybody reproduces, and they don't reproduce at the same rates, and that's because there's sexual selection in the diploid phase. And so of those that are able to reproduce, who gets to reproduce, is we can kind of think, and this is Darwinian sexual selection in the diploid phase, and this occurs intrasexually. So for example, male-male competition is an example of this, and intersexually, where there's mate choice. And in most species of animals, females are the choosier sex, but this influences of those that survived and can reproduce, who actually gets to reproduce and contribute gametes to selection in the next phase. So once gametes are produced, we also have selection on those. And most people don't even think about this. Like in a long-lived mammal, this part basically takes decades, and this part is, well, it depends on the species, this is at most days type of thing. So, we don't really think about this as being important, but it is. It's a little hard to conceptualize natural selection and separate it from sexual selection on haploid phase selection in animals, but we backtrack a second, it's much easier in plants. So for example, if you think of a fern which produces a gametophyte that's growing and living from a spore, and it produces sperm and eggs, the survival of that gametophyte is clearly natural selection in the haploid phase. Well, the egg survival and fertilization capability and sperm survival and fertilization capability are also natural selection in the haploid phase. So of the gametes that got produced, which ones can fertilize? Of the ones that can fertilize, we have sexual selection in the haploid phase, and this is post-mating, pre-zygotic sexual selection. No one knew this existed until the 1970s. So we have intrasex, which is sperm competition. The father of sperm competition, Jeff Parker, was here and got an honorary degree from us a few years ago and gave 2 seminars in the department. Until the 1970s, we didn't know this was a thing. But when we have sperm, so here males are competing for mating opportunities, Here, males are competing, the sperm of males are competing for fertilizations of a given set of eggs. It also occurs intersex through something called cryptic female choice. So when we have sperm from multiple males competing to fertilize a given set of eggs, females can actually bias that. They can alter the outcome of that and change paternity. And that's what's called cryptic female choice. It's cryptic because the male didn't know what happened, right? In most cases, the male knew if he got laid or not, but he doesn't know what happens to his sperm after, type of thing. And that's why it's called cryptic female choice. Now, so this summarizes selection in 4 of these stages across the life cycle. Well, it's actually not quite as simple as that. And this is what this figure is meant to incorporate. So the 4 corners here, this is exactly what I just presented. But we have kind of some linkages across these episodes and phases within one generation that can influence the outcome of selection. So the first one up here is quite intuitive. This is, Darwin knew about this. So it's the trade-off between sexual selection and natural selection in the diploid phase. So we take the classic peacock example. Male peacocks are males, but peacock with a big showy tail attracts more mates. This is what the females prefer, that's sexual selection. But having that big showy tail is not good for surviving being killed by a tiger, and so there's a trade-off in traits that are good for natural selection and traits that are good for sexual selection. That's within phase. Conceptually, there could be a trade-off between natural and sexual selection in the haploid phase as well, but that is totally unexplored. We also have connections across phase. We don't really conceptualize them as being a trade-off because they don't go back and forth because that switches generations. But for example, the experiences of an adult in the diploid phase can change the quality, for example, of its sperm, which alters selection in the next phase. So for example, your diet, for example, can impact your sperm quality. So if your sperm were in competition with another man's, this can influence paternity. You can also conceptually have the experiences of the haploid phase, what the sperm or eggs are doing prior to fertilization, and that may influence offspring development and performance in the diploid phase, and therefore affect selection in eggs. And I've become really interested on these linkages across these potential episodes for selection, and we try to zone in and try to actually get at these individual components. So the research approach that we do is kind of twofold. One is I measure things, and I used the caribou example earlier, but no different than if one of Eric's people went out and measured caribou calf survival or growth rate. Someone was out in the field and measured growth rate of a fish, or they were measuring Yolanda was doing growth of lichens or something like this. It's no different whatsoever. So I'm measuring things in the diploid phase, usually adults, because I'm interested in reproduction, their status or their quality. So for example, we might measure the age, size, shape, color, health, or something of diploid individuals. But I also measure gamete quality. So instead of just like how big something is and how fat it is or something, we actually measure the quality of their gametes. But it's just a natural situation. We do a lot of manipulation. So we experimentally manipulate things. So what we do is we experimentally manipulate the fertilization microenvironment, the exact conditions upon which fertilization takes place. We alter it, but we also manipulate the incubation conditions of embryos. It's a lot easier to manipulate embryo incubation than it is, for example, juveniles or teenagers or adults or whatever. So it's just a practical thing that I mostly focus on the early parts of development in the diploid phase. We do this using experimentation called split ejaculates, where we take a semen sample and we split it and we do different things with it. Or, more commonly in the literature, split broods, where you have sibling embryos that you then split and you put them under different conditions. So we monitor sperm and offspring performance when we do that type of thing. And in reality, we're often doing both. So for example, we might influence, we might measure how parent fish health status or something influences how sperm behave to different conditions in the haploid phase. So we're manipulating what the sperm are doing, but we might tie it back to what the adult experienced. I've worked in many different systems, but most of the work in the past number of years, there's kind of two main study systems. These things are wild fish spawning at different times of year. We have beach spawning capelin and hybridizing salmonids, and I'll explain why we do different things with different ones as we go along. So I'm going to walk you through some examples of research that we do and have done that identify some parts of these different components. So some of them I'll mention very briefly, some of them I'll go into detail. So first of all, let's just think about what is intuitively things that people think about the most, and that is selection in the diploid phase. So here's an example with capelin. Capelin spawn on the beaches. If you haven't experienced seeing this, you should. The eggs instantly stick to the substrate and they're fertilized within 2 seconds, and the embryos develop in the intertidal zone over 2 to 3 weeks before they hatch. There's no protection or anything, they're totally exposed to the elements. Here's a picture of some developing capelin embryos. They're cute little things. The dark spots here are their eyes, and the body there is curling around the yolk. These are almost ready to hatch. It depends on the temperature, obviously, but 2 to 3 weeks. When these things are incubating in the intertidal zone, lots of things can happen, but as an example, many of these beaches have a small stream, and depending on how much rain happens while they're incubating, you can get tremendous salinity gradients over small spatial scales along the beach. And I became interested in, well, does that matter? And so we did some split brood experiments. This is all in the diploid phase, where we created males and females, artificial fertilizations, take sibling eggs, split them, incubate them under different salinities. If you were going to do that with juvenile capelin, that would be very hard, basically impossible. If you were going to do it with caribou, you couldn't do it. You do it with embryos, you can do it in a beaker. And you basically put these in an incubator. We can have thousands of beakers potentially. The scope here is quite large, particularly with fish, because they're generally highly fecund. There's lots of embryos you can split from a batch. So we incubate these in the lab and done a bunch of different stuff, but here's an example of some work that would have come out of that and how incubation salinity and temperature, which I didn't mention, influence embryo development. Purely diploid, natural selection in the diploid phase. Similar context, some other work with former HQP. So the top here was with salmon on model predictions from a climate change on how winter temperature fluctuations might change, and we did work on how that might affect salmon hatch timing, and worked with Heather and Aline on banded killifish on temperature and threshold constraints upon which reproduction couldn't happen. But again, conceptually, this is all just natural selection in the diploid phase. It just so happens that we're narrowing in on the first few weeks of life. So all of that would have been in the context of over here. And again, that would have been context here in stage 1. What I want to give now is another example of some of the work we do, is how the diploid experience of the parent influences, for example, the quality of the gametes that are exposed to selection in a haploid phase. So diploid experiences on selection in the haploid phase. So here's an example of work that just got published just after Christmas. This was done in Ontario, It's a very complicated story. I'm not going to get into the details of it. I could give a whole seminar on this on its own, which people were interested in, some other context would work. But in this case, we looked at survival rates of old and young lake trout, which is natural selection in the diploid phase. But then we also looked at how the age of male lake trout influenced the quality of their sperm, which is then related to the ability of sperm to compete and get paternity in the haploid phase. In Atlantic salmon, we've been doing all kinds of work, and I'll dive into this system here now. So, Atlantic salmon are probably the most within a population, the most diverse vertebrate on the planet. And the related brown trout would be the most diverse vertebrate on the planet across populations. This species encompasses basically all of the life history variations seen in 7 Pacific salmon species. So it's very hard because of that to generalize. But just to get us started here, salmon are freshwater fish because they have to reproduce in fresh water. For example, their sperm won't swim in salt water. They spawn in the fall. Atlantic salmon spawn in the fall. The embryos incubate under the gravel in a nest that the female made. They incubate through the winter. They hatch very poorly developed to something, the jargon here is an elven. But when they've developed enough to feed in their first summer, they're called a fry. In subsequent summers, they're called a par. Some of them change physiologically and become smolts. Newfoundlanders call these salmon peel. And they go to sea. They go to sea for X amount of time where there's more food, but predation is higher. They fatten up, and the ones that make it come back to spawn. We'll come back to this story later, but that's in a nutshell what they do. But there's tremendous diversity in this. So this just captures some of it. For example, there are resident forms. Salmon, the life cycle is tied to fresh water. It is not tied to salt water. Resident forms never go to the ocean. And we have what are called waninish, which Newfoundlanders pronounce wininish. And these are both sexes, never go to the salt water. They're usually associated with growing up in lakes. Populations that are, quote, landlocked have to do this, but even in places where there's easy access to the ocean, they do this. Other resident forms are males that have an alternate reproductive tactic called precocial males. Females like big males. That's who they choose to reproduce with. But it's very costly to go to the ocean. You're likely going to die. So some males cheat, and they mature at little tiny precocial males. Females won't spawn with them, but when she spawns with another male, these guys go in and they chuck sperm into the mix all over the place, and they can steal a lot of the paternity from the big male. So the anadromous fish, the ones that go to the sea, come out, these are the ones people think about when they're thinking about salmon, right? These are the sea-run ones. But for example, in Atlantic salmon, there's highly variable age at which they go to the sea. It generally depends on the productivity of the environment in fresh water, but it ranges from about 2 to 7 years. So they can be 2 or as late as 7 years old before they go to the ocean to grow. Newfoundland, it's mostly 3s and 4s. Once they go to the ocean, there can be variable amounts of time spent on the first trip in the ocean before they come back. Some only go a few months. These come back and they're called a post-smolt, and they spawn the same year that they went out to the ocean. Some go for 1 winter. Generally it's 14 months. They're called a 1 sea winter. The jargon on that is called a grilse. By far, most of the salmon in Newfoundland do this. They come back and spawn for their first time after spending 14 months in the ocean. But there are others, when they went to sea, they stay out for more than 1 year. Of the ones that do that, most it's 2 years or 3 years, but in some places as much as 5 years, and they come back and they're this big. And they're also first-time virgin spawners. Unlike Pacific salmon, Atlantic salmon are not simulparous. They can survive and reproduce again, and they can do that many times. If they spawn successively, sequentially year right after the next, that's called a consecutive repeat spawner. They didn't skip a year, but some have skipped years, and so those are called alternative repeat spawners. But because they can spawn many times, you can have a fish that spawns once, and then it consecutively spawns, and then it skips 2 years, and then it spawns again, and then it skips 3 years, spawns again. So that on top of this, like in a given spawning year, there's an incredible amount of diversity in the age and history makeup of what the spawners actually were. Incredibly complex. We've been doing work on 2 aspects of all that diversity in turning how the diploid experience influences potential for selection in the haploid phase. Part of it is because of just practicality. These are things that I actually can work on and get something useful out of. So virgin grilled salmon are the predominant of the fish that anglers are catching here in the summer. The vast majority of the salmon coming up our rivers are this. So these are fish that left rivers as 2, 3, 4, 5-year-olds, as juveniles. Again, Newfoundlanders call them peel, but smolts in generally May. They went to the ocean and fed largely on invertebrates. This becomes important. They migrate long distances. These fish go up off of the Labrador Sea. They stay at sea for about 14 months. They come back to their home river, entering generally in June to July, where they've got a lot bigger, and they cease feeding. When they enter before they even enter freshwater, they don't eat anymore, and they spawn in November. Consecutive repeat spawners are fish that did that the previous year, but then they survived November spawning. They remained in the river another 6 months, so they didn't eat a thing for about a year. They migrate back to the sea in May, when they're called kelts, but Newfoundlanders call them slinks. When they go consecutive spawners, when they went back to the ocean, we know that they feed largely on fish, probably capelin. The successful ones hit a school of capelin, fatten up, turn around and come back. Because they stay coastal, they return to the river after only 2 to 3 months back in the ocean. They fatten up quickly in that 2 to 3 months, but they basically gain nothing in length. So they go out almost like a broomstick and they come back like a football, but they really haven't grown any. And they spawn in the consecutive November. So those are consecutive repeat spawners. We haven't done any work on the other forms. They're just rarer and we just can't really get access to them. From other people we know, for those 2 types of spawners, that they have different migrations and diets. So for example, Ian Fleming has been involved with migrations with those. And we know that the diet impacts carotenoids, because animals can't make carotenoids, and that those carotenoids are important. And I knew, for example, from years with this stuff, that there's variation among salmon that are returning in how the color of their flesh. And we knew when we started working on salmon reproduction that there's variation in the color of eggs among females, but we really didn't know if these things were linked or for similar reasons or not. So the hypothesis that developed from that was that these repeat spawners have lower stored carotenoids in their flesh because of diet than the virgin spawners. And that in females, this is manifested as repeat spawners have poorer quality eggs, and males having duller coloration and poorer quality semen. So to get at this question, we do work in, this doesn't show it well, but in central Newfoundland, where I grew up, on the Exploits River system, which is the largest river system in Newfoundland, but particularly because of a facility that's on it that allows us to get access to fish. The town of Grand Falls is called Grand Falls because it's located on the Grand Falls. It was put there because of a hydro plant to make the paper mill. There was nothing there before they put the paper mill, and they put the paper mill directly on the waterfall. That waterfalls is a complete barrier to salmon migration. But for about the past 35 years, there's been a fishway around the Grand Falls. And this is illustrated, there's two, this is the lower one. The salmon, this is totally artificial. The salmon enter here, they go up through a ladder system, they go up through here, they go out, and then they go up to another one. This facility is the Salmonid Interpretation Center, where you can go watch the salmon swimming through the ladder. But what we do is we take salmon out of this ladder. That's how we get access to our salmon. We take them out of the ladder. So some sampling of these salmon for these questions. So we had female salmon that we sampled in 2014, 15, and 16, and some of you will remember Brittany, who was a student working on this. And we were sampling males in 2016 and 2018, and this was with Marco and Madison. And when we get these salmon, I didn't come up with how you do this. The DFO does this all the time. You can take the scales from the salmon, and just like you can age a tree, the tree rings, you can age a fish with rings, but you can get a lot more than that. So you can actually determine the spawning history of the salmon by the patterning in its scales. So when we do this, typically we're, of the spawners, 10 to 15% of what's spawning are consecutive repeat spawners. And also, when you get spawners and you look at females, this is an example of the variation you see in the egg color. Some are quite orangish. They're not really red, but they're kind of orange, and some are very pale. And we didn't know at the time, but we now know that, just as predicted, these eggs belong to consecutive repeat spawners, and these eggs belong to virgin spawners. And it's so easy to tell. So for example, here's a standardized volume. This is a chicken egg yolk color chart, but it's very clear. And I can, with about 90% accuracy, if you give me a jar of eggs, I can tell you if the female is a virgin or a repeat spawner, because we've actually done that. So we actually do that technique now to plan our experiments, because we usually don't get the scale readings until months later, but based on the egg color, we plan our experiments, and then later we get a confirmation for which ones we were correct. Those eggs vary in color, but they also, of course, as expected, they vary in chemistry. So this is just some fatty acid, but for example, here's 2 years, virgin spawners and repeat spawners. You can see there's very clearly differences in the chemical composition of the eggs of the two types, which is likely related to the quality of them. In males, again, we think repeat spawners have lower stored carotenoids in their flesh. From that, we would think that repeat males have duller coloration during spawning and poorer semen quality. So, back to our diagram of the salmon life history, salmon are out to sea, they feed on fish, which is very poor in carotenoids, or they can feed on crustaceans that are very high in carotenoids, and again, our virgin fish are predominantly feeding on this, our repeats are predominantly feeding on this. When they come back, that's how salmon get their red color in their flesh. Well, what do they do with it? Well, females put it into their eggs. They produce orange eggs. Males put it into their skin, because sexual selection in the diploid phase operates because females choose males that are redder. If you're a redder male, you're more likely to get more mating opportunities. There's also work from other species that carotenoids are important in semen quality. So we're thinking that we know that the repeat females have duller eggs. We think that the repeat males also have a problem. So this is a salmon, male salmon in his full November tartan. Most people have never seen what a spawning Atlantic salmon look like. They look totally different than they do in the summer when people are angling them. They've got these beautiful patterns. And I can tell by looking at him now, based on this work, that he is a virgin spawner. So here's some work from Madison. So virgin spawners, there is variation, but you can even tell in this room that the repeat spawners, as predicted, are much duller in color. We look at their semen quality, and it seems to be fine. So on the left in these plots is the virgins, on the right is the repeat. So if we look at the semen density, so the proportion of it that is sperm cells, there's no difference. There's really no difference in the proportion of the cells that it will swim, and there's no difference in how fast the sperm will swim. So in terms of the competitive ability of the sperm, there seems to be no difference. So to summarize that, the virgin and repeat fish had different diets prior to spawning. We think that this relates to different stored carotenoids. Other people have figured out that that's true. We just haven't measured it in our individual fish. The repeat females do have duller eggs that differ in chemistry and are likely poorer quality, which would impact the offspring quality in the next generation. And the repeat males have duller sexual coloration. This likely leads to less mating opportunities, sexual selection in the diploid phase, but they seem to maintain sperm quality, which would be helpful under sperm competition in the haploid phase. All right, so moving on a little bit. So if we just think about it a little bit more simply, selection in the haploid phase, I'll give you a few examples. So we've done a bunch of work related to this in the species. This is some work with cod and how sperm swimming quality is affected by the ovarian fluid of farmed females. We've done work with Caitlin where we looked at how oil spill, oil pollution, and dispersants used to treat oil pollution change the abiotic environment. That changes the natural selection landscape of what these sperm have to encounter. And so it affects the natural selection in the haploid phase. We've looked at how salinity that the sperm swim in influences natural selection in the haploid phase. We've done a bunch of stuff with ovarian fluid in the context of cryptic female choice. I'll get in a minute. So we've done some stuff with, this is work with brown trout, salmon, and lake trout. This is work with Caitlin. So there's lots of things that we've been doing related to natural selection and sexual selection in the haploid phase. One thing that I've become increasingly interested in is with cryptic female choice. So I introduced this earlier, but again, this is an example of post-mating sexual selection in the haploid phase, where we get sperm competition, and cryptic female choice is a modification of the outcome of sperm competition. So she biases the paternity. In internal fertilizers, like birds, mammals, insects, there's many ways in which she can do that. But in external fertilizing animals, like most of the marine invertebrates and most fish and some amphibians, she, is not physically in contact with the sperm. But they do release chemicals with the eggs that we now know changes the sperm swimming characteristics. And if a female can change the swimming characteristics of different males to different degrees, then she can kind of give like one male a pat on the back and put her hand up on the other one type of thing and change the outcome of sperm competition. And that is what cryptic female choice is. And that's how it happens in an external fertilizer, including benthic in marine invertebrates like mussels. When we think about cryptic female choice, we usually conceptualize that as something that's intraspecific. So the female has spawned, and there's multiple males releasing sperm, they're from the same species, and so she's just biasing one male to the other male. That's the default way to think about this. But there can also be interspecific sperm competition. If the species are really totally different, it's a moot point, the sperm can't fertilize. But when fertilization is possible, often you can have multiple species of males spawning at the same time, competing to fertilize those eggs. If the female can bias the paternity towards her own species, that form of cryptic female choice is called conspecific sperm preference. And by doing that, she can reduce hybridization of her eggs. She has no control over who let the sperm go. So, if we think about hybridization, we have to think about what causes a hybrid. Obviously, you have to have both species breeding in the same place at the same time. You have to have a breakdown of courtship barriers, sexual selection in the diploid phase, that prevents mating. You have to have the release of gametes. You have to have fertilization. You have to have hatch, which is natural selection in the diploid phase. And you have to have survival long enough to catch a fish, but it could be context for any hybrid in the wild. So, if you went out in nature and you surveyed some population and 10% of the juveniles are hybrids, all of those things had to happen, but a bottleneck anywhere along the way will influence what you see. If, for example, there's strong sexual selection in the diploid phase where they won't breed together, well, you won't find any juveniles. But if there's lots of breeding, but some bottleneck later on, then you will maybe see some juveniles, but you have really no idea how much mating occurred. There could be all kinds of hybrid matings happen, but you don't see it because somewhere further down the chain is where the bottleneck happens. We have been working with hybridizing salmonids, mostly in the genus Salmo. So this is the genus that includes Atlantic salmon and brown trout. And both of these species are native to Europe. And they co-occur in the same rivers. They do hybridize, but it's a problem. It's an evolutionary dead end. Some of the hybrids are viable, but none of the grand offspring are viable. So it's a total evolutionary dead end when they hybridize. What we know, based on other people's work, that when Atlantic salmon and brown trout spawn together in sperm competition, that the female can exhibit conspecific sperm preference and biased paternity towards her own species. And she does that by the ovarian fluid that she releases with the eggs. That ovarian fluid upregulates. The sperm swim faster in ovarian fluid than they do in water, but she upregulates the sperm of her own species more than the other species. And it works both ways. Trout upregulate trout sperm more, salmon upregulate salmon sperm more. And this reduces hybrid fertilizations under threat of hybridization. Brown trout were introduced to North America, including to Newfoundland in 1883, and the frequency of hybrid juveniles in North America is higher than it is in Europe. Something is breaking down in this chain of all the things that can happen. In North America, where salmon are native, they do not create hybrids with native brook trout. I'm calling brook trout are a char. I'm going to call them char so we can use salmon, trout, and char to represent three different things. But here in Newfoundland, we have native salmon, we have native brook char. They don't produce, you cannot find a hybrid in the wild. But we have no idea how often they hybrid mate. And somewhere in that chain of what has to succeed, the barrier occurs. We don't know. Invasive brown trout do hybridize with our native brook char. And the offspring are called tiger trout, and they're sterile. But they do hybridize and produce something you can go out in the Waterford River and you can catch these things. With a hybrid mating, females have more to lose than males for a given hybrid mating. It's an evolutionary dead end for both, but a female produces like one batch of eggs a year, and most only ever produce one batch of eggs. So if that batch of eggs is hybridized, she loses everything. Versus a male can spawn multiple times a day. So a given hybridization event is more damaging for a female than it is for a male. And because of that, when she spawns, if she can avoid hybrid fertilization, it would make sense if she should. So how well can cryptic female choice mitigate hybridization? So this was work that Tyler did. Tyler just finished in the fall. So again, the frequency of hybrids in the wild is a poor predictor of hybrid mating frequency. In polyandrous matings with multiple males, in external fertilizers, these can release sperm at the same time. So internal fertilizers, when there's sperm competition, usually mating is not happening at the same time. One male inseminates the female, later on another one does, later on another one does. But in external fertilizers, it all happens at the same time. But all but one of those males was not the chosen mate. Female can choose when she decides to let the eggs go. She likes that guy, okay, she's gonna spawn with him. But when she does that, she has no control over who else lets sperm go as well. Cryptic female choice is therefore the last line of defense to prevent eggs from being hybridized. And the question is, how good is it? So again, coming back to our picture, we have a spawning group. The female totally controls when spawning is happening. She has total control for when she lets her eggs go, and she will usually only do that in the presence of a dominant male. But when she does that, other dominant males can come in and chuck sperm at the same time, and they do. But also, I mentioned the precocial pair. These little guys are adults. And they can be from her own species, they can be from the other species, and when she spawns with him, they're chucking sperm into the mix too. She has no control over the ability to prevent them from releasing sperm. But potentially she can control what those sperm do. So one of the things that Tyler did was this neat little experiment. This is in review now in evolution, but this is a split ejaculate experiment. So we take basically the ejaculate of a given male, we split it into aliquots, we swim some of the sperm in water, this gets at a baseline to male quality, then we spin some of it in char ovarian fluid, some of it in salmon ovarian fluid, and some of it in trout ovarian fluid. So the hypothesis is that char ovarian fluid upregulate char sperm more than the other species. Salmon ovarian fluid upregulates salmon sperm the most, and trout ovarian fluid upregulates trout sperm the most. This is a clear prediction if she can exert cryptic female choice, all three species. So here's a little figure showing the phenotypic plasticity approach of the sperm swimming in water versus an ovarian fluid. And this example, the species of ovarian fluids have just been averaged. This is the percent of the sperm that swim. This is how fast they swim. Each line is a male. See, there's some negative ones, but most of them are positive. Basically, if it has a positive slope, the ovarian fluid has upregulated the sperm. And it does. The key question is the relative amount of upregulation. So this is the money shot here. So the top panel is the percent of the sperm that swim. The bottom panel is how fast they swim. The clear prediction is that char ovarian fluid should upregulate char sperm the most, and they don't. The clear prediction is that salmon ovarian fluid should upregulate salmon sperm the most, and they don't, but they do in Europe. And the clear prediction is that trout ovarian fluid should upregulate trout sperm the most, and they do, but it's extremely minor and not worth talking about. So these are results that we were not expecting. And we have some ideas about why that may be, but I'll just move on to another topic here. So this example, that experiment, was something based on here in the life cycle, how a mechanism of cryptic female choice with ovarian fluid can bias the outcome of sperm competition. So that would have been plain work that would have occurred here at stage 4. But what I'm going to move on to now lastly is how what these sperm, for example, experience, no change in paternity, and how that might influence selection in the diploid phase. So haploid experiences on selection in the diploid phase. I got really interested in this when I read this paper. It's on Atlantic salmon, and what they simply did is that they have an ejaculate, they split it, one male, one female. If the sperm swim in water for various amounts of time, the offspring develop differently. So in a human context, thinking about, cryopreservations and sperm bank and all that, can you influence the offspring development in that way? But just having the sperm swim for various amounts of time before they reach the egg changes offspring development. There's no change in who the dad is. So sperm that swim in water for different amounts of time create offspring that develop differently. And there's two potential ways that that could happen. One is that all sperm in the ejaculate are affected. So some epigenetic thing is happening by swimming, and therefore changes gene expression of the offspring. Or There could be that, for example, you've got 100,000 sperm, instantly there's 100,000 sperm available to fertilize the eggs, but if you waited 30 seconds, maybe there's only 10,000 sperm left alive, and maybe what survived is not random. And therefore you've got a subset of the gene pool from the one father available for fertilization. So this was based on how long the sperm swim. So I got thinking about, well, if how long the sperm swim matters, maybe what the sperm swim in matters. And so the hypothesis is that the environment that the sperm swim in before they hit an egg influences offspring development. So offspring development differently if the sperm swim, in the context of our previous example, conspecific ovarian fluid or heterospecific ovarian fluid. So coming back to this diagram again, if there's multiple males, sperm are always swimming through ovarian fluid. And when you get a hybridization event, we know the offspring develop differently, but you can't really tease apart how much of that is due to the male genetics, or, for example, what the sperm did. And so this design is designed to tease that apart. So how did the sperm change? So Tyler did this as well. This is set up as a split brood and split ejaculate experiment. And we're looking at phenotypic plasticity of embryo development, but not depending on the embryo conditions, depending on the sperm conditions in the previous phase. So we're manipulating the sperm swimming medium, and in this case, sperm are swum in conspecific or heterospecific ovarian fluid. And this is a little conceptual design. We did this with salmon moms. We only really needed salmon dads to get at the question, so if we have the sperm species is the same, we have 3 treatments here. They swim in only water, they swim in salmon ovarian fluid, or they swim in trout ovarian fluid. So for salmon sperm, swimming in salmon ovarian fluid is conspecific ovarian fluid. Swimming in trout ovarian fluid is heterospecific ovarian fluid. But we repeated this with trout sperm. And when you do that, who's heterospecific and who's conspecific switches. So it's just a repeat of that. But by doing this, we could kind of scale or standardize the effect of swimming in conspecific versus heterospecific against the effect of having a different species of father. So it's a way of just the relative comparison here. So this is, the experiment is really a split ejaculate experiment. We're not really changing anything with the embryos in this case. So we have salmon eggs. We manipulate basically the water that we add the sperm to. It has salmon ovarian fluid or trout ovarian fluid. We mix it, we pour it on the eggs, we incubate them. We look at a variety of things related to diploid development. So for example, if you take the eggs after a few weeks and you put them in vinegar, it stains the embryos, and so these lines are fertilized eggs and these are unfertilized eggs. You can look at the fertilization success. But in Caitlin incubate for 2-3 weeks, salmon incubate for 5 months. We do that in these recirculating salmon incubators where there's water running through this. This is done in a cold room in the biotech building. We're setting up to do this in the new building here, although there's problems with this building for that, but this building has a facility for me to do this here. But that's how we do this, and for months we have to incubate these embryos. But you can put thousands of them in one of these things. And we look at what the diploid offspring do. I'm not going to get into the nitty-gritty of any of this, but just to show you one graph. So this is the length at hatch, something that's important for survival later on. These are all salmon moms. If they had a trout dad, they hatch much bigger than if they had a salmon dad. So a hybrid trout develop faster. Hybridized eggs hatch faster and they hatch more developed. But what we swam the sperm in really doesn't matter at all. So the experiences of the sperm in terms of conspecific, heterospecific ovarian fluid or no ovarian fluid, which would be unnatural, doesn't matter. But what this means is that females can use these egg cues to change paternity without some negative consequence through epigenetics or something influencing the offspring later on. And no one has ever done that type of thing before. I'm going to end here by contextually similar, and so it's upcoming project here, going back to Caitlin. Caitlin are spawning in the intertidal zone. I already told you that in the diploid phase, the embryos are sensitive to salinity. So embryos are very sensitive to changes in salinity. But is it possible that sperm exposure to variable salinities can prime embryos and make performance in the diploid phase better? This is something that's kind of hot in the literature now, not with Caitlin, but this idea that sperm exposure can actually improve conditions of the offspring. And again, this can happen by epigenetics. So swimming in low or high salinity changes some gene expression later on, which results in the offspring doing better under the matching considerations, or haploid selection, where only a proportion of the ejaculate makes it through, but there's something about the ones that made it through that is different. And Ranjan, who's in the back of the room here, is a new master's student who has just arrived and is doing this work this summer. So my last slide, this is a split ejaculate, split brood experiment. So we take the ejaculate of a Caitlin, we split it, we swim some sperm, for example, at 25 and some at 35 practical salinity units. We fertilize sibling eggs with them, then we split the brood, and we incubate the embryos, diploid conditions under 25 and 35. And the prediction is that the embryos, for example, will do better at 25 than if the sperm previously experienced that. And the embryos will do better at 35 if the sperm previously experienced that. So that's it. So to acknowledge the team over the past quite a number of years here, collaborators from different parts of the world and a whole different variety of funding sources. And so that's it for today.
So if anybody has any questions, I'd say just like throw your hand up and say it loud.
Yeah.
I was just wondering if, like, for the salmon, if females have different choosiness based on whether it's virgin or repeat spawner. I guess my question is kind of why, if there's an advantage to being brightly colored, why do the repeats start eating fish?
Well, yeah, so I think it's a lesser of evils. So that when these kelps go back to the water, they're not in very good shape. Most of them never make it back.
Okay.
But the ones that do come back, they have to fatten up really quickly or they'll be preyed upon, they won't escape stuff. And so, you're going out and literally, I think, particularly the frequency of repeat spawners is higher on the northeast coast, and I think that's not a coincidence with Caitlin. So they're literally going out early May, within, the Caitlin are starting to come in. Like, I think if they hit a school of Caitlin, like they've hit the jackpot. And they just feed, they fatten up, they hit some energy threshold and they come back. Some, presumably ones that don't gain that energy quite as quick, they go back to sea. Most of them don't survive, but the ones who do, they stay out for a whole nother year. They probably feed on a lot of variety of things and they come back much bigger. But they're so infrequent, we really can't sample those to compare. Right. So one hypothesis might be that the alternative repeat spawners might be some intermediate form, like they might be fine, but but the chances of surviving is so low that, so the dull ones, so this is the interesting thing about the sperm quality seems to be maintained. So, but again, they don't need to be chosen to release sperm. So, it would appear to be pretty clear that we think that the dull ones don't get as many mating opportunities. But by preserving their sperm quality, when another guy is where, they can chuck the sperm into the mix and, they probably, they'll get some proportion of the paternity and it maybe just be a lesser of evils, because, being choosy when you go back to the sea is not something that would normally pay off.
Right.
Yeah.
Is it known or possible to find out, do repeat spawners tend to have offspring who are repeat spawners?
We don't know, but there's no real reason to expect that, I don't think. However, maybe, it would be incredibly complicated to do that. These are not different genetic forms, but whether they went to sea for 1 year and came back, or went to sea for multiple years and came back the first time, that is under some partial genetic control. But the ones that make it, okay, so you're a female, and most of the repeat spawners are if you said, oh, there's 100 repeat spawners, most of them are females than males, and this makes perfect evolutionary sense. So the females have a batch of eggs, she produces the batch of eggs. If she survives to reproduce again, you know, she has to get those eggs. She can't make any more eggs in November. Like, it is what it is. But if you're a male, like, okay, you had a spawning event, well, why wait till next year to have another spawning event? Have another one after lunch. There is no lunch, they don't eat, but you know what I'm getting at.
Yeah.
Have another, you know, have 10 today, have 20 tomorrow. So it pays off for males to exert all of their energy and save nothing for survival, because the likelihood of going to sea and not getting eaten by a seal or a shark or something and making back is pretty poor. So if you can have multiple spawning events now, instead of saving something, it makes more sense. So the ones that do survive spawning are probably ones that didn't exert so much energy during spawning. So it may actually be, you know, not adaptive that they spawned at all. You know, it might have been that they never had as many mating opportunities or whatever, they never exerted so much energy, they got more reserved left. So, you suppose it's possible that, you know, there might be some inherited component to, you know, the proportion of what they could spawn in a season that they do versus holding. It's possible, I guess, that could be partially inherited, in which case the likelihood of repeating could be somewhat connected, but I'm not aware of any work ever on that, and I kind of doubt that it's actually the case.
So I think this is kind of in line with these questions, but look, what's the advantage in a stream where not the landlocked salmon, but the ones that kind of have this choice to go to sea or not? What's the advantage of going? Like, why do they go?
It's high risk, high reward, and what the real interesting question is conditions where that balance changes, and you can make predictions about the proportion that go or not, and can you find data that matches the predictions? So, females, the amount of eggs produced is totally related to how big she is. So her maximum fecundity is totally dependent on body size. So in our environments, basically the ocean is much more productive than fresh water. So she will undoubtedly be bigger if she migrates to the ocean to feed than if she doesn't migrate to the ocean to feed. So high reward, but the probability of predation is much higher in the ocean than it is in fresh water. So, whether she goes or not is this balancing act between, staying in fresh water and producing 50 eggs or going to the ocean and producing 2,500. Big difference for the females. But the males, and it often, as in many things, it boils down to frequency. Males have the ability to produce large numbers of offspring by different forms. Females will not spawn with only these guys. She will not choose them. So being a big male has advantages. Not only can you fight off other males and you can produce a lot of sperm, but she picks those. Lots of mating opportunities. But again, the probability of surviving, like in Newfoundland these days, the probability of a juvenile going to the ocean and coming back is about 4%. So, very high risk. So, if there's another way, it can pay off. This guy has a very high probability of survival from 1 year's, so this guy was, at age 4, he went to the ocean and came back at age 5, but 4% of them make it. This one at age 4, maybe 50% of them make it. But it has to do with the frequency, so if they all do this, doesn't pay off, because she won't spawn with them and they're in lots of competition with each other. But if this is very rare, it's a real winning circumstance. But it's also dependent on the probability of predation in the ocean. If predation is low, it makes more sense to go to the ocean. If predation is high, it pays to stay. And so of the fish that come up a river, most of them are female. Slight bias, but in these old age ones, we talked about, some go to sea for 2 years, some go for 3. The farther you get up in age, in terms of how long they stay, the more and more likely that is female. So almost all the ones that stay to sea for multiple years are females.
Okay, that was my follow-up question.
So because there's the relationship between body size and fitness is much more linear with a female than it is with a male.
Okay.
And so, yeah, it's a real balancing act. So, but like places where mortality at sea is higher than other places, you'd expect, mortality is higher, you expect a higher proportion of precocial parr. If mortality is lower at sea, you expect a smaller portion of precocial. But they all can't do it because she won't spawn with them. But they cheat. All right, well, thank you guys.